The 2 Weeks in the World web app can be accessed at twiw.genomicepidemiology.org. First off, we have the About page. This page will give you general information about the 2 Weeks in the World research project. There are resources available for download, and there are videos which you can also find on YouTube. Second off, we have the Explore Data tab. This tab will take you to the project dashboard, where there's an overview of the countries in which there are partners that are participating in 2 Weeks in the World. There's a sample counter showing how many countries and units are currently participating, as well as how many isolates and DNA samples, as well as the total count of samples received. There's a geographical distribution of samples, and there's also a genus distribution. Everything is shown according to the WHO regions. There's also a timeline for sample collection, which can be scrolled sideways. This timeline has bubbles indicating different numbers of samples collected at different times from different regions. In the top left corner, you can switch the dashboard to the analysis view. You can also switch back to the project dashboard on the same button. The analysis view is interactive, and this is where you can select countries. You can see the different types of species discovered in the samples that were received from this place. You can also see the different source types that these isolates came from. The species can be selected, and this will give you the acquired resistance of the species selected. Acquired resistance refers to identified resistance genes and the class level prediction of resistance for these genes. Third off is the phylogeny tab. In this tab, you can select a tree. The name of the tree will indicate the reference genome for this tree. When you select a tree, you'll be taken to a dashboard that shows you the geographical distribution of all the different samples that map to the reference, as well as a phylogenetic tree. You can zoom in and out on the phylogenetic tree. There is a list of all of these samples that are in the tree. You can select individual samples in the tree, and this will be reflected in the table on the bottom left side. Now for the sign-in option. Partners enrolled in 2 Weeks in the World will be given a username and a password for this app. When signing into the web app, you will be asked to respect the partnership of 2 Weeks in the World. Once you're signed in, the web app will look slightly differently, because instead of Explore Data, you can select a map or you can select a table. Selecting the map corresponds to the Explore Data tab. The table is only accessible to partners until all of this data is published and becomes open access. In the Tables tab, you can change the size of the font that you have in the table. You can also change how many rows you wish to see per page, and you can see what page you're viewing, and you can change this. You can then select different tables that you wish to view. You can search the entire table, and you can search individual columns. The first table, Overview of Samples Submitted to 2 Weeks 2020, will give you an overview of the different samples that we have received on 2 Weeks in the World and their pertaining metadata, both as submitted by partners and as validated by us. The second table, Sequence Raw Data Quality Control and Bioinformatics Species Identification. This table will give you the sequencing file name corresponding to a particular sample. The final dataset for 2 Weeks in the World consists of validated genomes. The genomes are validated based on the sequencing quality of that genome, but it's also being validated based on the species prediction. So we use K-mer Finder and we use RMLST to do our species prediction, and based on all of those three things we validate the genomes, and those data can be found in this table. The third table is bioinformatic analysis of acquired resistance genes. This table is a table that gives you resistance genes per phenotype. So every row is a sample. Every sample is only shown once, and every column is a phenotype. So there might be multiple genes identified with ResFinder that could confer resistance towards a particular drug. All of these genes may not all exist within this particular genome if the algorithm wasn't able to get a best hit. And next to every single phenotype, you will have a value. This is the match value. This is the same match value as you can see on the analysis view in the Explore Data dashboard. We then have something called ResFinder detailed data. This is in sorts the same information as is found in the bioinformatic analysis of acquired resistance genes. However, in this table, the same sample may occur many times, so there may be many rows for the same sample, but this time it will only be with one resistance gene per row. And in the columns, you have stats for that resistance gene hit according to the ResFinder tool. So you will have a percentage identity. You will also have a percentage coverage of that gene. Finally, we have the phylogenetic tree table. This data corresponds to the phylogeny dashboard, and you will have the different samples and the different phylogenetic trees in which they occur. We then have a research canvas for partners as well. So this requires signing into the web app. This is where you can submit studies. In order to submit a study, you simply give the study a title, you indicate which authors and institutions are involved in this study, as well as other partners involved, and this will be reflected by which samples are involved in that study. Also, you give some sort of description of your study, and then you click submit. For existing submissions, you can access these, you can read more about what they are, and you can comment on them. Finally, we have links to the Center for Genomic Epidemiology website. This is where we have online services in form of bioinformatic tools of many sorts that are openly accessible, free to use. You simply submit your sequence data and wait for your output. All of the different tools currently used on 2 Weeks in the World can be found here and are openly accessible. This includes ResFinder, it includes Evergreen, it includes K-mer Finder, and RMLST, which we actually also use for species prediction, is not hosted by us because we didn't create it, but it is openly accessible. Last but not least, there are currently shortcuts to signing up for the 2022 2 Weeks in the World study, and of course there's a shortcut to the metadata submission platform for partners submitting samples for 2022, and this is described in a different video. That's it. Have fun.